Welcome back, students. Today we are going to discuss Unit 5, and in this particular unit we are going to discuss sexual reproduction in flowering plant topic. So before I start the main content, I would like to inform that the images used in our slides are for education purpose only. We respect intellectual property right of the original creator and owner of these images. If you are the owner of any of the images used in this PPT, have any issue, any concern, please contact us at sathee@iitk.ac.in and we shall remove the images. In this particular video lecture, we are going to discuss these points: reproduction and development in angiosperm, structure and function of flower, development of male and female gametophyte in angiosperm. So, what is flower? Flower are not solely for human use. All flowering plant exhibit sexual reproduction, right? And diversity in inflorescence, diversity in flower and flower parts shows various adaptation of the plant according to their weather condition, their environment. And these adaptation actually ensure the formation of fruit and seeds, and the end product of which is the end product of sexual reproduction. Okay, so in this chapter we will cover the morphology of flowering plant, means flowering plants means angiosperm, and structure of flowering plants. And with these two topics, we are also going to discuss about the processes involved in sexual reproduction of flowering plant. Right? So flower, it is a fascinating organ of angiosperm. This is a general structure of flower. It contains so many parts like sepal, petal, peduncle, ovule, style, stigma, filament, anther. Okay, so this SSO, stigma, style, ovary, these three all together we can say female part of the body, or we can say pistil for the same. Pistil, female part of the body. We can further divide pistil into three sub parts: stigma, upper part, which acts as a platform for polar grain, and there is a longitudinal part which connects this stigma with the ovule, is known as a style, right? And this ovule, it contains reproductive cells which further develop into the seed after fertilization, right? And similarly, collectively we can use the term stamen for filament and anther, and stamen act as a male part of the flower. Okay, this longitudinal cylindrical part is known as filament, which actually support anther to make it accessible to pollination. And these double lobed structure is known as anther, which produce pollens, right? Like male gamete of a flower. Sepal, what is the use of sepal? Sepal actually protect the flower when it is in the form of bud as a outer covering, right? What is the role of this petal? This petal actually attract insects. Okay, insects, because they are bright in color, they are having a kind of scent or kind of smell, so they attract pollinators, means certain kind of insects, because of the bright color and specific type of smell. And this peduncle, it is a stalk of flower. So this is a general structure of flower which have different parts. Some part comes under female reproductive part category, some part comes under male reproductive part category. Now, what is pre-fertilization and what are those structures and events which comes under pre-fertilization category? So the decision of a plant to convert or to develop a flower, it occurs long before the actual flower appear. Okay, so there are certain hormonal and structural changes occur in initial differentiation and development of floral primordium. and in the form of inflorescence, some flower bearing different kind of flower bud, and eventually they convert into the flower. So what is androecium? Androecium actually consists of whole of stamen, which represents male reproductive organ, androecium. Another term is gynoecium. This gynoecium represents the female reproductive part. Right? So the decision for a plant to flower occur long before the actual flower appear. Now, one by one, we will discuss both the parts, male reproductive part and female reproductive part. So, first one is androecium, that is stamen. This is the stamen. So, a typical stamen has two parts. One is this anther and another one is filament. And this anther is generally bilobed structure. Bilobed terminal structure, and this is the long cylinder like stalk is known as filament, right? So the proximal end of the filament, this proximal end of the filament attached to the thalamus or petal of the flower, and number and length of the stamen vary across different species of flower. Some are having 5, some are having more than 5. So number and length, it depends upon the species to species. Okay. So anther, a typical angiosperm anther is bilobed in structure, with each lobe 2 theca or diathecus. We can use the term diathecus for the same. Okay, as it is bilobed in structure and each lobe having 2 theca, and a longitudinal groove often separate each theca, a longitudinal groove like this. So this longitudinal groove, okay, this actually separate each theca, and the bilobed nature is distinct in the transverse section of the anther. The anther is tetragonial in structure, means 4-sided structure. It is a 4-sided structure like this. 1, 2, 3 and 4, or 1, 2, 3 and 4. Okay, so it is a 4-sided structure, as you can see in this microscopic images also. So it is a tetragonial in structure in which 4 microsporangia are located on the corners. 1, 2, 3, 4. 4 microsporangia. Okay, and these 4 microsporangia is also known as pollen sac, right? And each microsporangia actually develop into a pollen sac. As just now I said, these are converted into the pollen sac, which extend longitudinally. Now it is extend longitudinally, okay, throughout the length of the anther, just like this. It extend longitudinally, and this longitudinal structure contains pollen grains inside it. Okay. Now, so sporangia develop into the pollen grains which extend longitudinally through the length of anther. Pollen sac, this is known as pollen sac, and these are packed with the pollen grains. Now, microsporangium. This microsporangium, if we cut it transversely, then in transverse section it appear nearly equal. Nearly not equal, circular in structure. As you can see, these are circular structure, right? Comparatively circular structure. So this microsporangium is surrounded by 4 different layers, 4 wall. Outer layer, epidermis. This one is the epidermis. Okay, followed by next this one, endothecium. Here this one, endothecium. Then middle layer, this one. Then middle layer. Okay, and then this inner, this is the middle layer, and this is the tapetum. and the innermost layer is known as tapetum. So it is a 4-wall layer, outermost epidermis, followed by endothecium, then middle layer, and then tapetum. Outermost layer, all three outermost layer actually responsible for the protection of the pollens. Okay, protection, they provide protection, and the innermost layer, which is known as tapetum, this particular layer actually provide nourishment to the pollen grains, nourishment to the developing pollen grains. And these cells have a dense cytoplasm and often more than one nucleus. Okay, and there are a kind of tissue known as sporangious tissue, which is a group of compactly arranged homogeneous cell present in the center of each microsporangium when anther is young. So another kind of tissue is present on the center, which actually present in the center of each microsporangium when the anther is young. Okay, now let's discuss microsporogenesis. Microsporogenesis is a process of forming microspores. Process of forming microspores from pollen mother cell or PMS. Development of microspores from the pollen mother cell through meiosis. Through meiosis. As you can see in this particular picture, this is a microspore mother cell. Via meiosis 1 from 1 cell to 2 cell, and then immediately second meiosis 2 occur. So it will further divide into another division. So from 1 cell to 4 cell or tetra will form. All 4 kind of cells get separate out, okay, and form pollen grains, right? And further a mitosis occur, and so nucleus is divided into 12. And now we can use the term mature pollen grains for the same. And this pollen tube or pollen cell contains outer layer known as exine, then inner layer known as the outer layer, then inner layer intine, and 2 type of nucleus, as you can see over here and in this zoom picture also. This is known as generative cell. Okay, in generative cell, generative nucleus is present, and here pollen tube nucleus is present. Okay, so this is the process of microsporogenesis. So microsporogenesis is a process of forming microspores from pollen mother cell through meiosis. So cells of sporogenous tissue undergo meiotic division in developing anther. Each cell of sporogenesis tissue can produce a tetrad, this tetrad, microscopic tetrad, acting as a potential pollen or microspore mother cell. Then this microspore are arranged in a cluster of 4 cells forming microspore tetrad. Just now you have seen that picture. So at that time an anther mature and dehydrated, and that time microspore actually dissociate and develop into pollen grains. Several thousands of microspore or pollen grains are formed inside each microsporangium, and it releases upon the anther dehiscence. Now pollen grains. Pollen grains, this is a zoom picture of a pollen grain. This is generally spherical in shape, and it measured about 25 to 50 micrometer in diameter. This is the measurement, 25 to 50. The pollen grain represent the male gametophyte. Pollen grain represent male gametophyte. It has prominent 2-layered wall, as I just now told you. Outermost layer is known as exine and innermost layer is known as intine. So exine and intine. Intine, it is a comparatively inner wall, it is comparatively thin and continuous, okay, which is composed of cellulose and pectin. Okay, outer one is hard. Now the outermost layer is Why it is hard? Because it is made up of sporopollenin. Sporopollenin. It is a most resistant organic material known. It is a most resistant organic material known, this hard layer, as it is resistant to the high temperature, strong acid and even alkalis. And no known enzyme degrade sporopollenin. And it contains germ pores where sporopollenin is absent. Only at this particular location, germ pore side. Pollen grains where germ pores is there, here only sporopollenin is absent. And because of the presence of sporopollenin in the outer surface, outer side of the pollen grain, these pollen grains can be well preserved as fossil. Some of pollen grains are still well preserved as fossil because of the presence of this layer, sporopollenin. And it exhibit various pattern and various kind of designs. Okay, now let's discuss the pollen grain structure and its development. Mature pollen grain contain two layers, not two layers, sorry, two type of cell. One is known as vegetative cell, another one is generative cell. Just now we have this kind of structure, na, outer layer, inner layer, outer layer, like this, and two type of cells, one here and over here. So one is generative cell, another one is vegetative cell. Vegetative cell, large in size with abundant food reserve and large irregular shaped structure. Vegetative cell size is big and it is having irregular shaped nucleus, while generative cell is comparatively smaller in shape, spindle shaped with a dense cytoplasm and nucleus. And this generative cell float in the cytoplasm of the vegetative cell. Over 60% of angiosperm pollen grains are shed at this two-celled stage only, in 60% of angiosperm. In the remaining species, the generative cell divide mitotically to give rise to male gamete before pollen grain are shed, means at three-celled stage. Okay, generative cell further divide into two. So, one nucleus is present in vegetative, one is present in generative. In 60% of angiosperm, remaining 40% further generative cell divide mitotically and produce from 1 to 2. So, three-celled stage. Just before the pollen grain are shed, they convert itself into three-celled stage. Clear? Now, pollen grain allergy. Pollen grains of many species can cause severe allergy to some people and severe kind of bronchitis or bronchial infection in some people. These afflictions often lead to chronic respiratory disorder like asthma, bronchitis in some people. A kind of grass which is known as parthenium, it actually introduced, which is this parthenium is known as, popularly known as carrot grass. This parthenium is introduced to India as a contamination with imported wheat, and it is a main cause or main cause of pollen allergy to most of the people who are actually sensitive or having this kind of allergic reactions. Okay, but pollen grains are having high nutritive values. They are nutrition rich. They are having a good amount of protein in it, a good amount of amino acid, vitamins, minerals, polyphenols, carbohydrates. So some people in some countries, pollen tablets are very much popular as a food supplement and widely available in Western countries also, mainly Western market, as a tablet and syrup. and some scientific reports and some kind of research paper claimed to if someone using these pollen grain syrup or tablet, that time these pollen tablets or syrup enhance performance of athletes and race horses. Now let's discuss about the viability issue of this pollen grains. As you know, pollen grain must land on the stigma for fertilization. Okay, but before losing and at that time when it land to the stigma, it must be at its viable form before losing viability for fertilization. So viability duration actually varies uh, species to species and as it is depends upon the certain kind of factors like temperature, like humidity. In some kind of cereals like rice and wheat, pollen grain lose viability within 30 minutes. Within 30 minutes. But in some other type of plant variety like Rosaceae, Leguminosae, Solanaceae, they remain viable for months. So that's why I told you it depends upon the type of variety of the plant or species type. Sometimes pollen grain can also be stored for years in liquid nitrogen at minus 196 degrees temperature for use in crop breeding program, just like some people preserve seeds in seed bank. Okay, now gynoecium. So this is all about the male reproductive part. Now gynoecium. This gynoecium is known as pistil and it is a female reproductive part of the flower. It is a female reproductive part as it consists of single carpel sometimes, okay, means monocarpellary, or multiple carpels means multicarpellary. Multiple carpels can be fused together and form syncarpus, or if they are multiple carpels are free, then we can use the term apocarpus for the same. So if they are free, multiple carpels are free, we can use the term apocarpus, and if they are fused together, that time we can use the term syncarpus. Okay, so each carpel have three different parts. Already briefly we have already discussed in previous slides, but here we will discuss detail about it. So it is having three part: stigma, style and ovary. SSO. Okay, stigma act as a landing platform for pollen grains. Here only pollen come or over here and land on the stigma, right? So stigma act as a landing platform for pollen grains. Style, it is a elongated part beneath the stigma, and then the ovary, which is a basal bulged part containing ovarian cavity or lacula. This is a ovary in which certain kind of ovules or megasporangium are present. Okay, so stigma, style and ovary. Each carpel we can further divide into three different subparts. Now, inside the ovarian cavity there is a placenta from which megasporangium or ovule arises. Clear? So, number of ovules in a ovary, it can vary from one to many. In some people, only one ovule are there, like wheat, paddy, mango. Some are so many ovules are present, right? Like papaya, watermelon, orchid, muskmelon, and so on. Okay, so it depends. So, ovule, megasporangium. Now discuss detailed structure of ovule. Ovule actually attached to the placenta. This ovule attached to the placenta by a longitudinal structure known as funicle from here. Okay, this attached to the placenta by the funicle. And there is another structure known as hilum. This is hilum. This is known as hilum. Okay, which is a junction between ovule and funicle. So this hilum is a junction. This is a junction between, this structure is ovule and this is funicle. So this hilum is a junction between this and this and this. So it is a junction. Clear? So this structure is known as funicle, and this is the hilum, which act as a junction between ovule and funicle. And then integuments, different kind of layers, different kind of protective layer, one or two protective layer encircling the nucellus. Okay, so this one is the outer layer, or we can say outer integument. This is outer integument, followed by inner integument. This is the inner integument. This is the inner integument. Okay, and then micropyle. Then micropyle, which is a small opening present at the tip of integument. So these are two integuments layer, and this is the small opening. This small opening is known as micropyle. Okay, and then chalaza. What is this chalaza? It is a basal part of ovule just opposite to the micropylar end. So micropylar end is present over here. So just opposite to this end means this part. This is known as chalaza. Clear? Now nucellus. Nucellus, it is a mass of cell which is present inside the integument. These are the integuments, outer integument and inner integument, and mass of cell present inside the integument is known as nucellus. Here this location is known as micropyla, just opposite chalaza, right? So nucellus is this area, mass of cell present inside the integuments. It contains abundant reserve food material. Now embryo sac. Embryo sac, it is located inside the nucellus. This is the located inside the nucellus, and it is also known as female gametophyte. Clear? So typically a single embryo sac is formed from a megaspore. Typical single embryo sac is formed from a megaspore. Now let's discuss Now we have discussed all total structure of female part of the flower. Now megasporogenesis. So megaspores are again formed from the, just like microspores, these are also formed with the megaspore mother cell, means MMC. So megaspore mother cell differentiate into micropylar region of the nucellus, and it is a large cell with a dense cytoplasm and a prominent nucleus. It is a large cell with dense cytoplasm and a prominent nucleus which undergo meiotic division to produce, meiotic division is there, then it will produce 4 megaspores from 1 to 4. Clear? Now female gametophyte development. One megaspore is functional, others degenerate. These are the megaspore mother cell. This is nucellus, this is megaspore mother cell. Only from 1 to 4 megaspore tetrad. Now we can use the term tetrad. Okay, so one megaspore is functional, other degenerate. The functional megaspore further develop into the female gametophyte, or we can say embryo sac. Clear? And this process is known as monosporic development. What is the process? What is the name of the process? Monosporic development. Nucleus of the functional megaspore divide mitotically. Okay? So nucleus of functional megaspore divide mitotically and form 2 nucleated embryo sac, 1 to 2 and then 4 and then 8. So 2 nucleated, then 4 nucleated, and then finally 8 nucleated cell. Right? So mitotic divisions are free nuclear division. So nuclear division occur, but there is no cell wall formation. No immediate cell wall formation occur. Okay, cell wall form after 8 nucleated stage. After when 1 to 2, 2 to 4, and when 1, 2, 3, 4, 5, 6, 7, 8. When this stage come, 8 nucleated stage, then only cell wall form to organize the embryo sac. Clear? Now, distribution within embryo sac. First is known as micropylar end, and this micropylar end contains 3 cells. 3 cells form the egg apparatus, and this egg apparatus, 2 synergids and 1 egg cell are there. 2 synergids, 1 and 2, these 2 are synergid cells, and 1 egg cell, this is the egg cell. And this is known as micropylar end. It contains 3 celled stage, 1 as a egg apparatus, 2 synergids. Clear? Another end is known as Chalazar end. This Chalazar end contains 3 antipodal cells. 3 antipodal cells, 1, 2 and 3. And in the central cell are present, which contain 2 polar nuclei. 2 polar nuclei. Okay, so again I'll count it 1, 2, 3, 4, 5, 6, 7, 8. Again 8 cells are there. 3 present over here as an antipodal cell, 3 present over here as a 2 as a synergids, 1 as a egg cell, and on the center part of the center cell, 2 polar nuclei are there. Okay, so in mature embryo sac, we can say this mature embryo sac are 8 nucleated but 7 celled structure. It is a 8 nucleated and 7 celled structure. Mature embryo sac are 8 nucleated and 7 celled structure, because in this cell, central cell, 2 nuclei are there as a polar nuclei. Clear? So 6 nuclei form cells, 2 remains as a polar nuclei. Synergids have filiform apparatus. These are the filiform apparatus to guide pollen tube. Okay, these synergid cells have this kind of apparatus also, filiform apparatus. So this is all about the today's video lecture. I hope most of the things has been cleared now. Still you have any query, any doubt related to today's video lecture, please let me know. We will discuss it later on. Till then, take care, bye bye, and keep learning. Namaskar.